Hello, we're glad you've joined us for this live webinar, StemoniX microBrain, 3D assay ready plates as a human ipsc-based neurospheroid platform for high-throughput drug screening. Today's educational web seminar is presented by LabRoots, the leading scientific social networking website and provider of virtual events and webinars, advancing scientific collaboration and learning. It's brought to you by Molecular Devices, one of the world's leading providers of high-performance bioanalytical instrumentation, software, and consumables, and is dedicated to helping customers unravel the complexity of biological systems. Let's get started. You can pose questions to the speaker during the presentation while they're fresh in your mind. To do so, simply type them into the drop-down box located on the far left of your screen labeled Ask a Question, and click on the Send button. Questions will be answered after the presentation. To enlarge the window, click on the arrows at the top right-hand corner of the presentation window. If you experience technical problems seeing or hearing the presentation, just click on the Support tab found at the top right of the presentation window, or report your problem by typing it into the Answer a Question box located on the far left of your screen. This is an educational webinar and thus offers free continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process to obtaining your credits. I would like to introduce Grisha Chandy, Senior Production Manager, High Content Screening Solutions at Molecular Devices, and he will be moderating this session.
Thank you very much. I wanted to just do a quick review of the agenda. I will introduce the technologies used in the seminar today and host the Q&A session at the end. Our first speaker is Cassiano Caramiu, lead scientist at StemoniX, who's going to talk about the development of the 3D human cortical spheroid platform for high homogeneity and functionality for high throughput and high content screening. So he's going to talk about how he developed this tool. Then we're going to transition over to Oksana Serenko, who is a senior research scientist here at Molecular Devices, who's going to talk about the application of this microbrain 3D model to a high throughput, high content toxicity and calcium oscillation assay. So I'm going to transition over and talk to you a little about the two or three technologies used in this presentation. The first is talking about automated imaging, and you can do imaging on a conventional microscope, and there's a manual process of putting your samples on the slide, putting a microscope, taking a picture, and then trying to figure out how to make measurements of that picture, and that usually takes you hours. With the Image Express system and high content screening, you can move those assays into multi-well plates or slides, but multi-well plates is what we're going to be talking about today. Put it into the system, acquire a variety of data types, automatically turn those images into numbers, and then try to make sense of the data you've collected. And that workflow takes minutes rather than hours. The complete solution for automated imaging is to acquire those data, those images, convert those images into something that can be segmented automatically by software, make these multi-parameter measurements, and then make sense of that data or do curve fits. And so that's the offering through Molecular Devices. The other technology that's being used in today's talk is high-throughput kinetic screen. So you put your samples in plates, you put into the flipper system, you add compound, you get tracings from all the wells at the same time, and you analyze that data and try to make sense of that data. Now, these two solutions have been combined with the products from StemoniX, where they are going to talk about their ipsc-derived microbrain, 3D microbrain model, and that's one example of it and a blow-up of what it looks like inside. And they've taken that product and put it through a variety of technologies, including the Flipper and the Image Express Micro, which are these high-throughput screening devices. So on that note, I would like to turn the presentation over to Cassiano, who can tell you about developing this 3D, the 3D human cortical platform.
Thank you very much, Girsha, for the introduction, and thank you everyone online today for joining online today. I'll be talking about our latest development, that's a 3D model cortical spheroid platform for high-throughput and high-content screening. So at StemoniX, our expertise is in human iPS-derived neural and cardiac platforms. We focus on adapting this platform for high-throughput screening and producing assay-ready solutions to accelerate drug discovery. So basically, the customer receives the plate ready to go to the assay. The cells are already plated, or in the case of the 3D, you have these spheroids in suspension for these assays. I'll be focusing my talk today on the neural platforms that we call microBrain, more specifically microBrain 3D. So one of the main challenges in neuroscience research and drug development is to find the best model to answer a question. This figure here summarizes the main models for neuroscience research. They vary from low complexity, such as cellular models in vitro, to high complex but low throughput, such as animal models or human post-mortem brains. Given the ethical barriers with the animal models in vivo and the scarce availability of post-mortem human brains, scientists have relied mostly on in vitro models such as cellular models and organoids. With the advent of human pluripotent stem cells, scientists now have access to any human tissue in vitro. The challenge, though, is to find models that recapitulate the key aspects of the tissue or the disease in vitro. More recent research have focused on the development of 3D models, which more closely resemble the human brain, for example. However, some challenges still persist, and between these challenges for the 3 models, the 3 main ones are: 1, how reproducible the assay is; 2, how accurate it recapitulates in vitro what you observe in vivo; and finally, how scalable this is. So here at StemoniX, we decided to tackle this problem, developing a new platform that's called microBrain 3D. So one of the main features of the microBrain 3D is that this is derived from a single donor iPS line. So both astrocyte neurons that is composed this platform is derived from a single cell line. This platform is composed of a co-culture of a balanced amount of astrocytes in neuron. You try to target 1-to-1 ratio of astrocytes in neuron, and they display key neuronal and astrocyte markers. We also observe spontaneous synchronized activity over time. So it's a sign of maturity of the system and connectivity between these neurons. And finally, we develop a platform that's amenable for high content screening. So all the platforms that we offer right now are in the 384 well format. On the left here, you can observe one cryosection of this microbrain of one of these spheres and highlight the neurons in green as they stand for MAP2 and the astrocytes in red as they stand for GFAP. One of the first things that we decide when we started to derive a highly homogeneous platform was to derive platforms that are highly homogeneous in size. When we talk about the 3D, there is a lot of variability that's in the system, and one of the hardest to control is the size of the spheroids. So we tackle these problems offering 3 to 4 well plate with 1 spheroid per well, and each spheroid is highly homogeneous in size between them. So across the plate, there's a lot of consistent in size. The range of these spheroids are around 600 micrometers, so we're not too big, so there is no hypoxia observed in the center, and they're not too small, so we can recapitulate many of the features of the human brain. When we receive this 384 well plate, we're able to culture for weeks. So here's just represented a snapshot of the whole 384 well plate. We capture this image using the image express microconfocal from molecular device. You can see one of these spheroids highlight on the right. On the bottom here, is the maximum and minimum representation, the graph, for the columns of the 384 well. So there's 24 columns in the 384 well plate, and you can see how compact is this distribution of the size of these spheroids. Of course, when we're talking about the 3D, also one of the main questions that you have, it is how you can translate everything that, or all the assays that you have in 2D to 3D. So it's very easy to do common assays in 2D, like immune staining. But in 3D, add some complexities that a lot of researchers are afraid of incorporating in their pipeline. We decided to tackle this problem in developing simple assays, or translate simple assays to the 3D platform. So one of the first one was immune staining. So as is highlighted here, this is a common immune stain with 4% PFA done in suspension. We use the Image Express microconfocal microscope to capture the images. So this is a Z-stack, is a Z projection of a Z-stack. But all the staining was done in suspension without the use of any special reagents. It's just a regular immunoreagents that we have in the lab. Moreover, the capture of these images was also done in suspension, so we don't need to mobilize or cross-section these spheroids to capture some relevant information from this platform. What I'm going to show now is a video of this platform now, also captured through the Image Express confocal, a time-lapse of the calcium oscillation.
In this video, you can observe the spontaneous calcium activity present in microBrain 3D. It was captured using Image Express microconfocal at 50 frames per second. Both images correspond to the same spheres, with the only difference between them being the color profile. This was done to better visualize this calcium oscillation that's happened.
As you observe in the video, you could see that there was synchronous activity or synchronous oscillation of this spheroid over time. When you measure the regions of interest of these spheroids, you can see that several cell bodies in these spheroids present this, neuronal cell bodies present this activity of calcium oscillation. Moreover, they are synchronized over time. So there is synchronous signal of these calcium oscillations that's being captured in 3D. We decided to ask if instead of using a low-throughput microscope like confocal that we're imaging one sphere at a time, if we could capture the oscillations that we observe in a more high-throughput fashion and use this, the flipper from Molecular Device. So using the flipper, now we can capture the oscillations of this calcium that we observe in the 3D in real time for the whole 384 well plate at the same time. Here highlights on the left is one well, and you can see the oscillation over time, so the change in fluorescence over time. This is recording made at 0.5 frames per second for 10 minutes. And you can see that from time to time we have this oscillation. Moreover, when we investigate the whole plate, all the wells that have spheroids, they present these activities, and this activity is very homogeneous between the different wells and between different plates. We decided to test if this activity correlates with electrophysiology. So we plate some of these spheroids on multi-electrode arrays, and we obtain and observe a lot of these oscillations happen on the multi-electrode arrays as well. So this correlates with the number of oscillations that we measure on Flipper. So what we're observing in Flipper, when you see these oscillations happen on the calcium, what we're observing is this massive synchronized signal that is going through the whole sphere. It means connectivity. We have a time of maturation that these signals start to happen in these spheres, but when they start, we're observing all across the whole plate at 384, and moreover, very homogeneous between the different wells. Of course, if you're talking about neurons, you're talking about cells that respond to a lot of chemicals, especially neuromodulators. So we decided to test if what you have is this signal that we're observing in Flipper can be modulated with some known neuronal modulators. Our culture is composed mainly of glutamatergic and GABAergic neurons. This is a cortical differentiation towards this cell phase. So on this first slide, I highlight some of the glutamatergic modulator. So we have a glutamatergic agonist like the KNX acid. And the KNX acid, you can observe that it increases the oscillation. So what you're showing here in the bar graphs, this is the peak frequency or the oscillations that you observe relative to wave control that you have. So 100% means no change in the oscillation pattern, and less than 100% means that we're decreasing the number of oscillation or repressing that signal. More than 100% means that we're increasing the number of oscillation or accelerating this oscillation pattern. And the KNX acid do just that, as is expected from an agonist of the glutamatergic system, but also start to repress after some time. We believe that at that point you start to reach excitotoxicity. So the stimulus of the KNX acid being too much, and instead of having continuous acceleration, you're having a repression because you have inducing excitotoxicity. The same neuromodulation was observed with glutamatergic antagonists. So we have several classes of antagonists represented here: CNQX, NBQX, DNQX, and NK801, and also then reduce these activity, reduce these oscillations in a dose-dependent manner. So more concentrated, more the reduction that we observe. Our system is also composed of GABAergic neurons. So we decide to test if we can have also GABAergic regulation there. We test the GABA agonists, baclofen and muscimol. So these are compounds that are going to potentiate the GABAergic system. That's the inhibitor system. Once you are potentiating that, you are reducing the number of oscillations, and you are able to capture that also in a dose-dependent manner. There is a lot of interest of our platform for seizure approach, and we decide to test if non-seizogenic compounds can induce some change in this platform. So we test the 4-aminopyridine, a non-seizogenic compound, and also induce increasing oscillation over a in a dose-dependent manner as well. So, of course, many of our customers and the people that collaborate with us, they're not used to these readings of in the flipper. These spontaneous readings in the flipper, especially in 3D. So it's hard to correlate between multiple platforms. So you use here the flipper, the image express confocal, and the multi-electrode arrays to see if the changes that you observe in the platforms are similar. Multi-electrode arrays is now the standard for electrophysiology, and a lot of people understand quite easily what are the changes there. So here on the bottom you can see vape control treated and 4-aminopyridine, that is a seizogenic compound, and you can see that there are clear changes in the profile and the pattern of activation of these neurons. But these changes are also observed on the flipper. So on the top here we have the vape control, the same vape control versus the 4-aminopyridine treated wells. And you can see the change in pattern. So there is an increase in the frequency of these oscillations, similar to what we observe in multi-electrode arrays. Finally, you also observe that in Image Express confocal. So Image Express, now we're doing a high-resolution imaging time-lapse of these cultures using the confocal microscopy. And one of the beauty of using Image Express and the flipper is that you can use back-to-back. This particular experiment, we use the same vape control that we measure in flipper. Right after measuring the whole plate in flipper, you can go to the Image Express and do a high resolution only of that spheroids that have interest. For example, there is a modulation, a specific modulation that you have particular interest in. You can record that using now the Image Express confocal to see these oscillations and to confirm the data. Once you have this powerful platform in-house, you decide to use in several projects and case studies here. This is just to mention some of the examples of how we are using in-house here. For example, for assay development and disease modeling, using especially for modeling neurodevelopmental disorders, we start to Rett syndrome in-house. We also doing for drug discovery. We have a customer that we're doing with some drug discovery for epilepsy, drug discovery of a library of compounds that can modulate the nervous system. We also did recent with a collaboration high-throughput screening of the low-pike library. That's a library of 1,280 compounds. And you find very interesting hits and a lot of reduced variability between the response from well 12 and the replicate. So that was really nice. Finally, we're also using for safety pharmacology and toxicology. So I'm going to show one example of neurotoxic screening for drug candidates to treat Zika infection. And Oksana, following my presentation, also going to show some examples of that, of collaboration that's going really nice for molecular device about environment toxins and their toxic effects. So Zika virus have been in the media for some time. There was a lot of attraction worldwide, especially because of the consequence. So it was causing, especially in pregnant women, some birth defects. It was a new virus identified in the ways that the infection in new locations, and especially in Brazil, it hit hard with a lot of people getting infected. The vector there is endemic, so it spread really fast in Brazil. And although there was a lot of effort from the scientific community to speed up the drug development, there were no current vaccines that were available or effective medications for Zika virus. So one of the approaches that the scientists decided to take, it is to repurpose drugs that are already FDA approved. So instead of start the pipeline from zero and develop new compounds, let's screen the library of compounds that are already approved by the FDA, so you can repurpose these compounds and put to the clinic much faster than a new compound that's need to test for efficacy. One of the problems, though, was to identify compounds that were not only approved but safe to use, especially in pregnant women. So this is a very particular case that if you're using a compound as prophylaxis to avoid any consequences of the Zika virus, we need to make sure that that compound is safe. So we need to make sure that it not induce any toxicity, especially in the central nervous system. So we decided to test that with the platform that we have in-house. The first test that we did was a tox profile, a cell viability profile of this. We use two different stages of our cell lines. We use a stage that was progenitor stage, that we call here NPCs or neurospheres, and you use also a more mature stage that's for more mature neurons, and we call microBrain 2D and microBrain 3D. So we have the two platforms in 2D and 3D. We decided to test both of them to see how is the efficacy between the two platforms and how they differ. And there was a lot of correlation between the two platforms. So we're very pleased to observe that you can mimic the finds that you have in 3D, in 2D, in the 3D environment, especially using techniques that are very common in the industry, especially for toxicology. The only difference between the two is that instead of using the cell titer globe per mega for the 2D, you use the cell titer globe per mega 3D for the 3D platform. And you can use any plate reader to read this assay. In this case, you use the SpectraMax from molecular device to do these readings. One thing that was very clear, there is a lot of compounds that induce toxicity, especially at high doses. So what you're seeing here is a heat map of the cell viability. And more gold means that's less cell viability, more blue means 100% viability, so no change in the cell viability. And a range of concentration of 5 different concentrations of compounds, from 10 macromolars to 0.01 macromolars. And you can see that in the highest concentration, especially in the progenitor stage, that's the NPCs and the neurospheres, there is a lot of toxicity of these compounds. Not surprising, many of these compounds that are being repurposed to Zika, they were approved for cancer treatment, and especially targeted cell cycle of the cells. And for progenitor stage, the cell cycle is really crucial and important for that cell line. So that's why we believe that we observe so much toxicity at the progenitor stage, but not too much toxicity in more mature stage or when you have more post-mitotic neurons. One thing to keep in mind, though, is that if you're using this in pregnant women, you need to be aware that that can influence the fetus development as well. So you need to avoid these compounds that incur too much toxicity at that stage. We decided to select some of the compounds that we consider safe, so the compounds that didn't induce much toxicity, to test in a functional level to see if, even though they don't have a cell viability drop, if there induce any functional change on these cell lines. We again use both platforms, microBrain 2D and microBrain 3D, but now we use the flipper tetra again. So it's a more high-throughput screening of these compounds. The difference between the 2D and 3D, the way that we do, is that usually when we use the microBrain 2D, you are induced a response to glutamatergic agonist. So you induce that artificial response with the compounds that will stimulate the cells. In this case, they're using the glutamate itself or the kainic acid. And what we want to observe is that on that stimulation, when you hit with the compound, after the pretreatment with these compounds that were considered safe in the cell viability, if they induce any change from the control or the treated cells. And we did observe 3 compounds that did induce some change, in fact, on this. So they were, although they didn't induce too much cell toxicity, they did induce a functional toxicity that could be observed on the flipper. Moreover, when we test on the microBrain 3D, because the microBrain 3D has this spontaneous signal, we don't need to induce that response. So what we're doing here is just measuring the spontaneous signal that we observe. And we pre-treated these compounds to measure if there is any interference of that. Surprisingly, we did find similar results from the 2D, but also found that there is a couple of compounds that did induce change in the peak frequency or in the frequency of that peaks compared to the microBrain 2D that could not capture that difference. So basically, microBrain 2D, with the induced response, we didn't capture change that could observe the microBrain 3D with the spontaneous oscillation. So there was a couple of compounds, especially the ones highlighted here, pyrimethamine and the palonosetron. You can see that the highest concentrations, there is a changes in the considerable change to the control compared to the controls. While in the microBrain 2D induced response, you didn't observe that. Moreover, you also found compounds that were relatively safe, like enricasan, that's one of it was highly coded for treatment of Zika virus. Finally, decide to see if you can measure this difference that you observe on the 3D, microBrain 3D using the flipper, also capture in the electrophysiology standpoint. And we're really pleased to see that these compounds that were safe in the microBrain 3D, like the enricasan, didn't induce any considerable change in several parameters that we measure using a microBrain 2D in multi-electrode array. So this is electrophysiology on the left. So changes here in the electrophysiology, they are or towards less frequent, that's going to be more white, or more frequent, that's going to be red. So we're looking for compounds that maintain this heat map close to blue. So that's why the control is the first one, and you can see that daptomycin and the enricasan also keep a pattern that's closely remembered to the control. But all the other compounds that were considered safe, we can see a safe in the cell viability. You can see that there is a functional toxicity. Moreover, we were able to capture that functional toxicity using the microBrain 3D. So the main difference here between the multi-electrode arrays and the microBrain 3D is because the throughput of the microBrain 3D being at 3 to 4 wells is much higher, especially in combination with the flipper. You can also see every time that you see this change, you can now take this change that you observe in the flipper and take this plate and measure more high resolution using the image express confocal. So you can even have a high resolution of that signal that is inducing some change that you think that's important. So overall, we think that our platform is very capable to answer very complex questions like this one and investigate very problematic problem like the Zika virus infection in a very fast way, especially in combination with the platforms from Molecular Device. So in summary for this webinar, what I hope that I show you today is that the microBrain 3D is a neurospheroid platform composed of mature cortical neurons and astrocytes. So they present highly consistent functional signal between the different wells and make it suitable for high-throughput screening. The neural networks responsible to neurotransmodulators, and we don't need to activate that signal. Is a spontaneous signal that arises from the development of this 3D that you can modulate with the compounds later on. And we can use this platform to investigate studies that can be applied at different levels. So immunostaining, as I show, cell viability, as I show with the cell viability, and finally functional. That's one of the key aspects of this platform to be used to. So I would like to end my talk. Just thank you, the StemoniX team, and leaving here some contact information for more information. And with that, I'm going to pass to Oksana, that's going to continue show some of the exciting finds that we have with the collaboration that we have with molecular device. Thank you very much.
Thank you so much for your introduction. So this is Oksana Sirenko. I am senior scientist at Molecular Devices. So I will be talking about evaluation of neuronal toxicity using stem cell-derived neuronal 3D cultures. So there is increasing need on using complex and biologically relevant, more predictive cell-based assays to screen and evaluate compounds with potential for neurotoxicity. The current methods for neurotoxic evaluation rely mostly on animal models, which have their limitation related to throughput, cost, and relevance to human data. So there is a pressing need for the development of multidimensional screening strategies that could provide improved predictivity and also will allow to facilitate prioritization of chemicals for in vivo testing. There is also very important is requirement for assays to be compatible with a high-throughput screening. So the goal of present study was to evaluate feasibility of microBrain 3D mixed neurospheroids for neurotoxicity screening, also to optimize phenotypic readouts for quantitative compound responses, and test a selected set of known neurotoxic compounds. We used fast kinetic fluorescence imaging on the flipper or imaging system to measure patterns and frequencies of the calcium oscillation. Spontaneous oscillation of neurospheroids were monitored by changes in intracellular calcium levels with calcium-sensitive dyes. Here you can see control spheroid calcium oscillation. We used the assay optimized for high-throughput screening in the 384 well plates. In a typical experiment, we received pre-plated spheroids from StemoniX. Then we cultured them for 4 days and then treated with compounds. In this example, you can see the screenshot from the 384 well plate on a flipper. Recordings were done for 10 minutes. You can see control samples and also samples treated with different concentration of drugs. Each well of the 384 plate contained single uniformly sized spheroids and generated highly consistent performance across the wells and plates. Neural spheroids were exposed to compounds in concentration response for either 60 minutes or 24 hours. The assay allows phenotypic characterization of oscillation profiles by using multiparametric analysis that include the oscillation rate, the frequency, the amplitude, peak width, distance between peaks, as well as peak rise and decay times, and where form irregularities. The analysis is done using ScreenWorks Peak Pro software used for peak analysis. Here we present example of traces of several neuroactive reference compounds that includes known drugs like lidocaine, lamotrigine, dilantin, and haloperidol. You can see dramatic changes in a peak frequency. You can see decrease in a frequency and also increase in spacing and irregularity. Also, we've tested set of known neurotoxic compounds, including methylmercury, rotenone, MPP, and berberine chloride. We also observed dose-dependent changes in readouts, including decrease in a peak rate, amplitude, also changes in the spacing and peak width. Changes in a peak frequency and amplitude were also observed with compounds with unknown mode of action, including flame retardants and pesticides. In addition to calcium flux assay, High-content imaging methods were used to gain additional information about morphology and viability. Spheroids were imaged using automated confocal imaging system, the ImageXpress microconfocal, with either 10x or 20x objective. The methods for imaging spheroids were previously described, but typically this stack of from 10 to 20 images taken separated by 5 to 10 micron, starting from the bottom of the well and moving up. All individual images can be saved and used for 3D analysis, or analysis can be done using 2D projection image. Here you can see spheroids stained for cytotoxicity assessment. Spheroids were stained with calcium AM for viability stain, mitotracker orange for mitochondria potential, and nuclear dye stain Hurst. The analysis for imaging readouts outputs were generated using custom model editor. The analysis first finds spheroids as an object, and then secondly defines number of live cells, which identified by presence of calcium AM signal, and then third identify number of cells with intact mitochondria stained by mitotracker orange. Dead cells could be identified by absence or decrease of calcium AM signal. So here you can see the masks for spheroids, also mask for positive cells, live cells stained with red, and negative nuclei stained in blue. So as you can see here, the control spheroid has a strong staining for calcium AM and mitotracker orange, and the one which had decreased viability have decreased number of live cells, and you can quantitate all these changes. In the present study, we characterized kinetic and also confocal imaging methods that compatible with automated instruments and demonstrated the potential utility of human ipsc-derived 3D neuronal spheroids for toxicity screening. Here you can see a list of phenotypic descriptors used for neurotoxicity evaluation. Multi-parameter analysis delivers informative readouts that enable screening for effects of test compounds on neuronal activity and also on cell morphology and viability. We've estimated the percent of variability for those readouts. We used control wells on same plate or different plates, just DMSO samples, and calculated the percent of variability. So for different readouts, this percent of variability presented here for 60 minutes or for 24 hours. As a next step, we use the method to evaluate toxicity effects of compounds from a library of 91 compounds received from National Toxicology Program. The library contains representative examples from different classes of chemicals, including pharmaceutical drugs, pesticides, flame retardants, and polyaromatic hydrocarbons. This work is currently in progress. Here you can see a heat map of the responses for the peak rate, where blue corresponds to decreasing peak rate and red corresponds to increase in oscillation rate. You can see that number of compounds cause significant changes in the peak rate and also in other readouts. In this example, for a number of pesticides, you can see how compounds can be ranked for their potential toxic effect. In the bottom, you can see highly toxic compounds like permethrin or deltamethrin. And in the top, you can see compounds with less known toxicities. We can calculate effective concentration of the compound effects for different readouts. And then we can run compounds for their potential neurotoxic hazard. On this graph, you can see effective concentrations for different readouts. So the smallest one presented. And as a black triangle, there is effective concentration for corresponding viability effects. So this information can be used for flagging potentially toxic compounds and for prioritization of those for other tests. This method is very suitable for screening of multiple chemicals in the high-throughput format. So in a summary, we were able to demonstrate that neurospheroids respond for neurotoxic effects and can be used for evaluation of neurotoxicity of various compounds. And it's very important that Assay is amenable for high-throughput assay format and can enable customers to do screening of multiple compounds. So I would like to acknowledge our collaborators from the National Institute of Environmental Health Sciences for providing this set of compounds for testing, and also collaborators from StemoniX and my coworkers from Molecular Devices. Thank you so much for your attention.